All right, guys. So today you're going to be getting a little bit of practice with Punnett squares. And we use these Punnett squares to sort of predict the outcome of certain traits for different offspring. So we'll take a look at what the genotypes are for the parents, so what their genes say for a particular trait, and then we'll look at the possible outcomes that we could have for their children, for their offspring. You guys are going to have a copy of this as well, and all of your work is to be done towards the last few slides. It does look like a lot, but it's all sort of like an introduction in the beginning, just notes through, like, I would say, the first 3/4 of this assignment, and then your work, like I said, will then be done on like that last those last few slides. Matter of fact, let's take a look here. Where will you be doing your work? Slides. So your work starts on slide 32. Okay. And you're going to work all the way through slide 39. So again, it may look like a lot, but I'm going to go through some examples with you to at least get you started. So hopefully that helps make things easier on you. And obviously you can skip around with this video to just sort of help you where you need it. So before we get into those examples, I do want to review a couple of important new vocabulary terms that we started. So I'm going to put this in presentation mode and we'll go from here. So some of the terms that you're going to see throughout this assignment are dominant. So remember, we have two different versions of traits or two different alleles. There's a dominant version, a dominant allele, which is masking the presence of the other version of the allele or of the trait, which is the recessive version. And the dominant version is always represented by a capital letter. The other version of a trait, if it's not a dominant version, then it is considered to be recessive. And this is when an organism with this trait will only show this trait when there's no dominant version of it present. So the recessive allele is represented by a lowercase letter, like you see here. And we'll go through some examples to really sort of, hopefully explain a little bit better what I mean by recessive only shows up when there's no dominant trait around. So here we have it, right? Dominant versus recessive. So we know that for each trait, we get one copy of our genes from each parent. So that's why our genotypes, okay, we have two versions of that trait together. So in the case where you see two dominant versions of the trait, so two capital letters in our genotype, okay, this is going to show you an offspring with the dominant trait or the dominant version of the trait. And in this case, it is straight hair. So it tells you that a capital T, the dominant version of this trait, is straight hair, and the lowercase t is representing curly hair, which would be recessive. So in the case of a genotype where you have a dominant and a recessive allele together, so a capital and a lowercase letter, you will see that the offspring still represents or still shows that it has straight hair. Again, because one of the versions of the dominant trait, that capital T, is there. In the case where you have an offspring whose genotype has two lowercase t's, two recessive versions of this trait, that's when you are going to see something with curly hair, showing that recessive version of the trait. And some of the terms that I was just using earlier, a genotype is describing the genetic makeup of an organism. So again, this is when we're talking about those different letter combinations. So you might have a capital, lowercase, a lowercase, lowercase, or two big letters together, or two capital letters. So again, genotype is just showing you the different alleles, right? Or the different letters. Whereas the phenotype is describing the physical appearance, what we're actually seeing, what characteristic are we actually physically seeing on this organism. So in the last example, we were talking about the hair type. Is it straight hair or is it curly hair? Other examples would be your eye color. Do you have blue eyes, green eyes? Are you tall? Are you short? Things like that. Right? Phenotype is the physical appearance. Okay, homozygous and heterozygous. And this is really referring to the genotypes. Okay, so if you have an organism that's genotype is homozygous, this just means that the letters in the genotype are the same. Whether those letters are capital letters representing a homozygous dominant, right? Because we know capital is referring to the dominant version of the trait, or you might have homozygous recessive, which is again homozygous. You have two of the same letter, and if it's specifying recessive, then we know it's going to have two lowercase letters or two little letters. Other than that, if you're not homozygous for dominant or recessive, then you would be heterozygous. And heterozygous is just showing that you have two different letters. Okay, so heterozygous, think different. So you have one capital and one lowercase version. And remember, when an organism is heterozygous, we have a capital letter there. We have a version of the dominant trait. Then the phenotype that we see from this heterozygous organism is going to be the dominant trait. So a Punnett square, as I mentioned before, is just something that we use. It's like a little puzzle to help us predict the outcome of future generations and what they may look like, what their genotypes might be, and then how might that reflect the phenotype of these organisms. So again, it's just giving us the possible outcomes, or the probability that an offspring will have that particular gene. So the way we set these up, and you'll see in a second, I'll break them down, how you fill them out as well. You get your genotypes from your parents. Okay, so we have, in this case, we have two heterozygous parents. So they have a capital A, lowercase a, and then another big A, little a over here. And we put their genotypes on the top outside and then the left outside of the Punnett square box. And you can see we separate them so that one letter is over each column here, and then one letter is aligned up with each row here. And then in the middle, okay, these are the possible outcomes or the possible genotypes for these offspring for this next generation. So, well, how do we complete it? So we already have it set up here, right? Again, we have our parent genotypes. One is split at the top, and then one is split on the side here. So now we need to fill in the Punnett square, fill in this puzzle, right? So the first thing that you need to do, and you can do the side or the top first. It doesn't matter what order you do them in, but I would recommend trying to just do either one or the other. Don't try to do them both at the same time because it could get a little confusing. So let's just say in this example, we're going to start with the parent genotype on the side. So you'll take this first letter, this capital A, this big A, and we'll fill it in to this first box here. Now you're going to take that same letter. Oh, not yet. I mean, you could, but anyway, now we're going to look at this bottom letter. Okay, this lowercase, this little a, and we're going to do the same thing. Move it into that box, that empty box in the middle. Okay, now you're going to take that big A from the top, and you're going to fill in to that far box, that far empty box, all the way to the right. And then you will do the same thing with the bottom A, the lowercase a. Okay, so again, just stick with one side at a time. I think that makes it go a little bit easier. But as you probably guessed, now that we've moved and put the gene or the different alleles in each of these boxes, now we're going to do it for this parent, for the one on the top. So we'll take this big A, bring it down into that box, and now you see we have two big A's here. We'll take this little a, bring it down to this top box, and you can see we're starting to fill in. And then same thing here. Okay, big A goes all the way down. So now we have big A, little a in this bottom box. And then the last step is you take that final allele and fill it in there. And here's your final product. Right, so we have the parents' genotype separated on the top and the side here, and then the possible outcomes for the offspring in the middle. Now, what exactly do these letters mean? These letters combinations mean? Well, again, they're predicting the possible outcomes for these offspring. So in this example, let's just say that the dominant version of the trait would be to have hair. So capital A is representing something with hair. A lowercase letter, the little a, the recessive version of the trait, is representing an organism with no hair. So when we break down the genotype ratio, which is just a fancy way of showing you how many have each type of each genotype, we will say that there is one big A, which you can see in this box here, two big A, little a's, so two heterozygous for their hair, and then one little a, little a. And then if we're looking at in terms of the phenotype, the physical appearance that these offsprings would have, um, three of them would have hair. And again, that's because three of these possible genotypes have that dominant allele that's representing an organism with hair. So anytime you see that dominant allele, it's going to show that dominant trait. And the one in this corner here that has two recessive alleles, the two recessive versions of this trait, little a, that's going to have no hair. All right. So at this point, I'm going to hit escape just like this says. And I'm going to take you through a couple examples of what you need to do on some of these slides just to get you started. So again, you can flip through these slides that I just went through at your leisure to help you along the way, and you can skip around this video too for wherever you need it. So in this first slide, okay, what you're going to do is you're going to tell me the phenotype based off of the genotypes that they give you. So phenotype, we're looking for the physical appearance. So what does this organism look like? So in this first example here, okay, our genotype that we're given is heterozygous for the R's. So we have a big R and a little r. So let's take a look at this table over here. R is representing body shape. So they either have round body shape or an oval body shape. But because we have that capital R there, it's representing the dominant version of this trait, this dominant version of this gene. So we go back here, we see the dominant gene for body shape is represented by a capital R. So the the phenotype that we would see here is just round. And if you want to specify round body shape, that's fine too, but I'll know what you're talking about. Okay. Now let's take a look at the next one here. Big S, although those S's kind of look upside down. But anyway, big S, big S. So you have two dominant versions of this trait. So let's take a look at the table at the top. We see that S is describing the pattern or representing the pattern of this organism. So we see that the capital or the dominant version of this trait, represented by the capital S, is striped. So then the pattern would be striped. And that is again, the phenotype, the physical appearance of an organism whose genotype is big S, big S. And now one more example here. You have little b, little b. Two recessive alleles. So that's fine. B on the chart here. Okay, B is representing the color of this organism. And since there are no capital letters, since there's no dominant versions of this trait in the genotype, then that means this organism will show the recessive version of this trait. In this case, it would be red, represented by the little b. So the color or the genotype for this organism would be red. And there you have it. So hopefully that's a good introduction for that slide. This next one, it's sort of the opposite. You're given the genotype, you're given the phenotype, and you have to say, what are the possible genotypes? And remember, if it's showing a dominant trait, that means it could be homozygous dominant, meaning the genotype has two capital letters in it, or it could be heterozygous, which would be one capital letter or one big letter and one lowercase letter. And again, that's because if anytime that dominant version of the trait is present, that's what we are going to see, the dominant phenotype. So in the case here, okay, let's take a look at this first one. It says oval body. So body shape, we're going to take a look. Okay, it's telling us the dominant gene is round. Oval is recessive. All right, so if it's oval body and it's showing us that recessive version of the trait, then there's only one possible outcome for the genotype, right? And that genotype, here we go, is little r, right? Two lowercase r's there. Again, you can't have a capital R in there or else we would be showing the dominant trait. So let's jump to this one right here on the right, curved fin. So fin shape, the dominant version of the trait is curved. So it's telling us that we have the dominant version of the trait here. So this would be one of those examples where you could have two possible genotypes because it's not telling us whether it's homozygous or heterozygous. We kind of need to leave that possibility up in the air. So for this example, the possible genotypes would be capital C, or capital C, lowercase c. And again, because a curved fin is the dominant version of the trait, it could be homozygous dominant, so big C, or it could be heterozygous, meaning that it has just one version of that dominant trait. Alrighty. And this one is just asking you to tell me if something is homozygous or heterozygous, this next slide here. So you feel free to go back into the slides to help you along with this. And you don't need to write the whole term out. If you just want to write HO for homozygous and HE for heterozygous, that's fine. So first two examples, let's do it real quick. We have one big A and one little a, so we have two different letters here. So this is going to be heterozygous. I'm just typing it out completely for you. But again, like I said, you can just put HE. I'll know what you mean for heterozygous. And then this one here, okay, we have two big B's. Since we have two of the same letter, whether they're two big B's or if they were two little B's, it's still two of the same letter. So that's going to be homozygous. Homozygous. And then again, you guys for this can just type out HO or HE. I'll know what you mean. All right. So last thing, I want to go through. So I think you guys can do these examples on your own. Let me take a look at this one together, okay? Because it's asking you to give me the genotype and the phenotype possibilities. So I'm going to fill out this Punnett square real quick for you, okay? But again, just a reminder, right? We take each of the alleles, each version of the trait, and just fill in the boxes. So boom, boom, straight across. So we'll go. Well, I like to do the capital letters first. So I'm going to do big B. So we're taking the top parent first. Big B, big B, and big B, because again, both types are the same. Big B, and now we're going to take the parent from the side, little b, little b, little b, little b. So that's how your Punnett square should look. So wouldn't you know it, all of these possible genotypes are exactly the same. So for this case, we would put big B, little b, big B, little b, and then we could say 4 out of 4. Or if you want to do percents, you can just say 100%. So you're just telling me what are the possible genotypes. And if this was a little bit different, let's say you had one that was big B, then you would just tell me big B, 1, and then so on. I'm just filling in the rest of them. Now the possible phenotypes. So again, you have the phenotypes at the top. Capital B, the dominant version of this trait, is brown eyes. Little b, the lowercase b, is blue eyes. So the recessive version is blue eyes. But because all 4 of these genotypes are showing the dominant trait, right, they have that capital B, then you're going to see 4 out of 4 brown eyes. And there you have it. Okay, so use that to kind of guide you in this last slide, number 49, or excuse me, number 39. But I think you guys should do just fine.